Then, with reference to the cell, on considering the cell, certain terms are there. When we tell about the osmosis, if the entry of water towards the cell, that process is known as endosmosis. If the diffusion of water outward from the cell, that kind of osmosis is known as exosmosis. With reference to the cell, we are using the term endosmosis and exosmosis. Then, usually the endosmosis takes place when the inward diffusion of water through the semi-permeable membrane occur when the surrounding solution is less concentrated. When the surrounding, if we place a cell in a less concentrated solution compared with the solution and the cytoplasm inside the cell, if we compare it, that if endosmosis takes place when outward solution is less concentrated. If we put the cell in a high concentrated solution, the water inside the cell will go outward, so that exosmosis will takes place. Then you can see that exosmosis that usually occur, you might have observed, if we put the tender mangoes in salt solution, or if we put the tender mangoes in salt, we can see that the cells in the mango will become shrink, so that that pickle we make with the tender mango, in all the mangoes will appears to be very small because exosmosis takes place. Then another thing we do in our daily life, when we see the snail, to kill that snail we put salt on it, so that the salt can absorb water inside the cell of that snail, so that snail may die. That we do daily when we experience such things in our daily life. Then again, another thing we learned, the exosmosis takes place when we put more fertilizer around the roots of plants, if it is summer season, or if we do not pour enough water, or if we are not watering the plant properly and putting too much fertilizers around the soil, around the root in plant, that plant will become destroyed because of the exosmosis. The salt or fertilizer inside the soil may absorb all the water from the soil, so all the water from the roots, so that plant may die. Then again, here you can see one experiment. Here you can see a kind of funnel known as a thistle funnel. Here inside the thistle funnel, a solution is taken, that is sugar solution, and the mouth of the thistle funnel is tied with a particular kind of paper known as cellophane paper. Instead of cellophane paper, we can take egg membrane or animal bladder. Egg membrane meaning that membrane, after boiling the egg, when we remove the shell, we can see a small membrane. That membrane can be taken without rupturing if we put the egg in dilute hydrochloric acid for a day. We can take that egg membrane without any rupturing. So such egg membrane or cellophane paper, that egg membrane is a semi-permeable membrane that is tied at the mouth of the thistle funnel. Then sugar solution is also taken. Then we submerge that part of the thistle funnel inside the pure water or less concentrated solution. The water compared to the solution inside the thistle funnel, that is sugar solution, that is a high concentrated solution, and water is less concentrated. Then you can mark an initial level. When we start the experiment, we can mark the initial level. After 2 or 3 hours, we can see the water level will rise inside the thistle funnel. Level will rise. So that from that experiment, we can understand that water may entered from the beaker to the thistle funnel. That is how the water level rises in the thistle funnel. Here another control experiment for the comparison. Experiment for comparison is known as control experiment. We can take another thistle funnel with plain water filled in it and suspend it in another beaker also containing water. Here you can see we cannot see any rise in the water level in the thistle funnel. From that we can understand that because of the high concentrated solution, the solvent molecule, that is water molecule, passed through the semi-permeable membrane. At the same time, even though it is higher concentrated solution inside the thistle funnel, that sugar cannot come out. If we taste the water in the beaker, that will not taste sugar inside it. From that we can understand that only water passed through the semi-permeable membrane and causing the rise in the level in the thistle funnel. Here you can see the 3 main conclusions from this experiment. The experimental setup: some water of the beaker has passed through the cellophane paper to enter the thistle funnel containing the sugar solution. Sugar from the thistle funnel has not passed into the beaker. The cellophane paper has acted as selectively or differentially permeable membrane. The same experiment we can do with the by taking more concentrated sugar solution in the thistle funnel and very dilute or less concentrated sugar solution in the beaker. We can see the same kind of experiment. Sugar solution, high concentrated solution inside the thistle funnel, and instead of water, less concentrated solution. We can use the same experiment. We can see the same result. Then again, the same kind of experiment again we can do with the rubber sheet. Rubber sheet is impermeable, so we cannot see the rise in the water level. Only if it is the membrane is semi-permeable, we can observe the rise in water level. Again, muslin cloth. Muslin cloth has very big pores, so that if we put sugar solution in the thistle funnel, the muslin cloth will allow the sugar also pass through the muslin cloth, so there will not be any water level inside the thistle funnel. Instead, the lowering of the water level may occur because the whole solution, because of the gravitational force, flow through the muslin cloth. So it is a freely permeable membrane. Then that are the other modified versions of this experiment. Then again we can see another term we have to learn is osmotic pressure. Here the definition of osmotic pressure. Osmotic pressure is the minimum pressure that must be exerted to prevent the passage of the pure solvent into the solution when two are separated by a semi-permeable membrane. Here you can see that if we keep the same experiment, you can see that here in this experiment, if we keep the beaker and the experimental setup, we can see a initial level and a final level. Level rises up to a particular level, then rising of the water level will stop in the thistle funnel. It is because of the gravitational force and the weight of the solution itself will prevent the further entry of water into the thistle funnel. If we keep this experiment for more time, up to a certain level, water level will rise. After that, that will stop because of the pressure exerted by the solution inside the thistle funnel, that will exert a force because of the gravitational force and weight of the solution exert a force and stop further entry of uh, uh, water into the thistle funnel. That pressure is known as osmotic pressure. We can measure it with by applying a weight. Here you can see a piston, a airtight piston is used here. A weight is placed there. We can measure the osmotic pressure. Osmotic pressure is the minimum pressure that must be exerted to prevent the passage of pure solvent into the solution. If weight were there, if we were not placing a weight there, water level may rise. If we put a weight, when we put a weight there, the water level will not rise. That is the minimum pressure or weight that we are exerting a minimum pressure there, so that will prevent the entry of water into the thistle funnel. But, that is also osmotic pressure is also a definition that may be asked in the examination. I think you understood it. Today I am finishing my class. Thank you for watching.